Hello and welcome everyone to our webinar on leptin and diabetes. My name is Corinth Auld. I'm Mercodia's Medical Science Liaison and the host of this event. We welcome any questions you may have, which can be submitted at any point during the webinar. I'll hold the questions until both speakers are finished, at which time I'll read the questions aloud and give our speakers a chance to answer as many as we have time for. So it's a great privilege to introduce you to our two speakers today. The first is Dr. Jeffrey M. Friedman, a physician-scientist studying the genetic mechanisms that regulate body weight. Dr. Friedman's research on various aspects of obesity received national attention in late 1994 when it was announced that he and his colleagues had isolated the mouse OB gene and its human homologue. They subsequently found that injections of the encoded protein leptin decreases body weight of mice by reducing food intake and increasing energy expenditure. His current research is aimed at understanding the genetic basis of obesity in humans and the mechanisms by which leptin transmits its weight-reducing signal. He is currently a professor at the Rockefeller University, an investigator at the Howard Hughes Medical Institute, and the director of the Starr Center for Human Genetics. Dr. Friedman was elected to the National Academy of Science in 2001. His work was referred to in Time Magazine's Best of Science section in 1995 and 1996. He also received Popular Science's Best of Science Award in 1995, was elected to the Royal Swedish Academy of Sciences as a foreign member in 2005, and has received many prestigious awards and honors such as the Endocrinology Transatlantic Medal from the UK Society for Endocrinology, the Bristol-Myers Squibb Award for Distinguished Achievement in Metabolic Research, and in 2013 was elected to the American Academy of Arts and Sciences. We are very happy to have him with us today, so without further ado, Dr. Jeffrey Friedman.
Well, welcome everyone. Looking forward to, together with Dr. Unger, speaking primarily about leptin and diabetes. But if you look at the first slide, you can see a brief outline of what I hope to cover today. I would like to first tell you a little bit about where leptin came from as a prelude to discussing leptin physiology and pathophysiology, and then segue into its use as a treatment, primarily right now for a form of diabetes known as lipodystrophy. This is a leptin deficiency state, one of several leptin deficiency states, and I'll talk about leptin's use in this particular condition, and then I'll close by briefly telling you about the relationship between leptin, obesity, and diabetes in the majority of type 2 diabetic patients for whom leptin is not as effective as it is for lipodystrophic patients. On the second slide, you see a picture of two boys. The boy on the left is massively obese. The boy on the right is quite lean. And of course, the question we began addressing was what accounts for this dramatic difference. Many possible explanations for why body weight can differ so dramatically have been suggested, and I'll go through a few of these in a moment, but first I'll point out that in addition to the cosmetic differences, obesity carries with it important medical complications. As shown on the next slide 3, there are a number of comorbidities associated with obesity: diabetes, hypertension, heart disease, and NASH. And in aggregate, these are the primary causes of morbidity and mortality in the developed world. These are also growing problems in the developing world. On the next slide, you can see the health impact of obesity. What's shown is the relative risk of death as a function of different BMIs, an indicator of adiposity. And if you look primarily at the magenta line for both women and men, what you'll see is that as BMI moves above 30, the relative risk of death increases both among men and women. Now, the question, in a sense, that many people might ask at that point is, if having an elevated BMI increases the relative risk of death, and also carries with it social stigma that lead obese people to make less money and be promoted less readily than lean individuals. Why don't people just lose the weight, move to the flat part of the curve, improve their health? Now, on the next slide are 3 general explanations offered up for why obese people have difficulty losing weight. Some individuals suggest that the obese lack a level of willpower that is normally exhibited by lean people. I find that point of view is more often favored by lean people. A second explanation is that obese individuals are so because of a modern lifestyle and environment that predisposes to obesity. And the third possibility is that there are genetic or biological differences between obese and lean individuals. Now, I think an enlightened view of this condition would require that we consider the possibility that all contribute, as is the case for any other disease. But what I want to briefly communicate now is that the evidence suggests that genes are the most dominant contributor to obesity. In particular, is the major contributor for morbid obesity. So the next slide simply says genes as a prelude for my showing you some brief genetic evidence supporting the role for genes and differences in body weight. On slide 7, you can see the results of twin studies. A simple way to assess the genetic contribution to a trait is to systematically compare monozygous and dizygous twins. If the monozygous twins are more similar for a trait, you can surmise there's a genetic basis. And on slide 8, is the result of such analyses showing that obesity is as or more hereditable than most traits that have been studied, with the exception of height. So on slide 8, it shows that obesity it gives a hereditability score of 0.8 to 0.9. This exceeds the hereditability of many disorders that we accept have a genetic basis, including schizophrenia, diabetes, hypertension, alcoholism, heart disease, and many others. To illustrate the power of these genes, I want to tell you now about a case report on slide 9, which shows an image of the boy I began this presentation by showing you. This boy was of normal weight at birth, but began to develop morbid obesity in infancy with extreme hyperphagia. He was already pre-diabetic at the age of 4 when he weighed 90 pounds. This child came from a highly inbred pedigree, which often suggests that the cause could be genetic. Now, Stephen O'Reilly and colleagues in Cambridge noted that this child had some similarities with an animal strain that we'd been studying, as shown on slide 10. This is the Obie mouse. The Obie mouse is obese as a consequence of a defect in a single gene. The result of that gene defect leads animals, as shown on the far right of that slide, to weigh 3 times as much and have 5 times as much fat as lean individuals. In 1994, we identified the Obie gene as encoding the hormone leptin, shown on slide 11. Leptin is the afferent signal in a feedback loop that maintains homeostatic control of fat mass. I'll show an animation in a moment that outlines or reveals to you how leptin works, but for the moment I'll simply tell you that leptin is made by fat and signals to the brain regulating appetite. In the Obie mouse, leptin was missing. As a consequence of this, the animal becomes fat. If you now look at slide 12, however, what you can see is that an Obie mouse given saline remains obese, while leptin replacement in an Obie mouse, as shown on the right, leads an animal to normalize his weight. So this was the origin of leptin. It was the defective gene in genetically obese Obie mice. O'Reilly noted this work, and as shown on on slide 13, he measured the blood levels of leptin in the young child I showed you a picture of, and now followed the outcome in this child similar to what was done with the mice after leptin treatment. And as you can see, the child loses weight despite getting taller. On slide 14 now is the longer-term consequences of this. It's a picture of the same child at the age of 3 and the age of 8. And what you can see is that the child's weight has essentially normalized, these are the same two images that I began the talk by showing you. So in this case, what was causing the child's obesity was not a lack of willpower or the modern environment, it was the lack of the hormone leptin. On slide 15 is an animation that I'll show you now that exhibits how the physiologic system works. At an individual's stable weight, the animation should go on now, fat mass is stably maintained, leptin level is made, and food intake equals energy expenditure. If weight is lost, which is after a diet, leptin level falls. A low leptin level is a stimulus to eat more, and an animal or a human who has a low leptin level will eat more until such time as fat mass and leptin level return to the starting point. Conversely, if weight is gained after a binge of eating, fat mass and leptin level will go up. At least in a normally responsive individual, this high leptin level will suppress appetite and again return weight and fat mass to the starting point. So by this mechanism, body weight can be maintained within a relatively stable range. And what's shown on the final still panel here is that essentially what this system does is provides a biological force that resists weight change in either direction. This system, in a sense, solves an evolutionary problem by making certain that the species is neither too thin nor obese, both of which have important negative consequences in the wild. Now, on the next slide, which is slide 16, you see a picture of the boy. This child is obese because he lacks the hormone leptin. Now, one of the important features of this phenotype is that the child is not only obese, but as shown or as stated on slide 17, has a number of pleiotropic other abnormalities in perhaps every organ system. And these abnormalities are not typically associated with obesity. Ironically, all of the abnormalities that this child manifests, or most of them, are actually normally associated with starvation. In addition to reducing body weight, leptin treatment also corrects these other abnormalities. And it's this conceptual framework that lays the groundwork for the use of leptin as a therapy. And that groundwork is shown on slide 18. When fat mass is lost, leptin level falls. And as I showed you, this provides a stimulus to the brain, acting on nerve cells in the hypothalamus to eat more. But in addition to regulating weight, a low leptin level activates what we might refer to as a starvation response, the net effect of which is to conserve energy. And so leptin-deficient children, much the same as starved individuals, show alterations in thyroid function, immune function, insulin signaling, and reproductive function, among many others. In aggregate, these various systems consume energy, and so the lack of leptin turns down, in a sense, these energy consumptive processes. Now, this in turn has set the stage for the uses of leptin as a therapy, because endocrinologists surmise that there might be pathologic states associated with low leptin levels that result in abnormalities in one or more of these systems. And if that were the case, restoring leptin levels to normal in these leptin deficiency states would lead to improvement of health. Now, on slide 19 is an example of this. This is a condition known as lipodystrophy. This is a leptin deficiency state and an important form of diabetes. Lipodystrophy is the congenital or acquired loss of adipose tissue. In a sense, it's the fat equivalent of muscular dystrophy. There is an alteration, developmental or otherwise, that leads to a loss of the organ. Because there's low fat mass, there are low leptin levels. And low leptin levels can be associated with insulin resistance. And so patients with this syndrome develop a severe, often intractable insulin resistance with diabetes and a fatty liver. As first shown by Shimomura, Brown, and Goldstein in Southwestern University, leptin treatment in animals markedly improves this condition. As a consequence, a number of investigators began to test leptin in human lipodystrophy, this loss of fat mass. And these included Dr. Simeon Taylor, Ali Foral, and Philip Gordon at the NIH, as well as Dr. Garg at Texas. If you look at slide 20, you can see now a patient with one of the forms of lipodystrophy. This is so-called congenital generalized lipodystrophy. These patients have either recessive or acquired forms and have no fat at all anywhere in the body. There are a number of genetic causes of this that are listed, and this condition has a number of important consequences that are shown in part on slide 21. If you look at the left panel, this young woman had a body covered with xanthomas as a consequence of very, very high triglyceride levels. She also had a hugely protuberant abdomen that was comprised almost exclusively of an engorged fatty liver. After 1 year on leptin therapy, the xanthomas disappeared and her liver had now approached a normal size. In fact, on slide 22 now is an image, an MRI image of her liver before and after leptin. On slide 23 are some of her blood parameters. This young woman with generalized lipodystrophy had poorly controlled diabetic ketoacidosis, nephrotic levels of proteinuria, and hypertriglyceridemia. And as you can see in these data set, all of these abnormalities were markedly improved or corrected on leptin therapy. On slide 24 is simply a reminder that leptin was approved a year ago for the treatment of complete generalized lipodystrophy. However, there are other forms of lipodystrophy, and this is summarized on slide 25, where it's pointed out that while leptin was approved for generalized lipodystrophy, it was not approved for other less severe forms, in particular partial lipodystrophy. On slide 26 is a diagram just illustrating the fact that there are different forms of this condition. Patients with generalized lipodystrophy have no adipose tissue. Patients with partial lipodystrophy have variable fat loss, usually conserving adipose tissue in the central regions, but with a lack of adipose tissue in the extremities. On slide 27 is an image of a patient with familial partial lipodystrophy, illustrating the fact that this patient had central obesity but skinny arms and legs. Partial lipodystrophy patients also in some cases appear to respond to leptin therapy with an improvement in diabetes, although the response is less robust than can be seen with the generalized patients. This is shown on slide 28, where the reduction in hemoglobin A1C on leptin therapy is shown for all lipodystrophy patients tested in NIH, as well as broken out for both generalized in blue and partial in red. And you can see the reduction in A1C is greater for the generalized, though there is a response for the partial. On slide 29, you can see the same thing now for partial lipodystrophy, for triglycerides, a greater response among the generalized versus the partial patients. And then on slide 30, you can also see that all these patients appear to have an improvement in their NASH score on leptin therapy. Now, overall, if you turn to slide 31, the patients with partial lipodystrophy respond less well to leptin than the patients with generalized lipodystrophy. And it was perhaps in part for this reason that the FDA, for the time being at least, has limited its use for the generalized form of the disorder. Now, there are two possible reasons for why the partial patients respond less well. The first is that in general they have less severe baseline abnormalities. In addition, they tend to have higher endogenous levels of leptin that seems to reduce the response. If you turn now to slide 32, what you can see is that partial lipodystrophy patients usually have more fat than do patients with generalized lipodystrophy. And if you click the animation, you can see also that generalized patients typically have lower leptin levels than partial patients. Now, we should now be on slide 33, which says that to some extent leptin level predicts the triglyceride response after leptin treatment of this condition. In general, if a patient has partial lipodystrophy, if their starting leptin level is below 4 or 5 nanograms per mil, it predicts a more consistent triglyceride lowering, but a higher level did not preclude a response. So in this case, each patient is represented as a line with their starting and final triglyceride level on leptin therapy. The same type of data is shown on slide 34 for hemoglobin A1C among these patients. Again, most patients, even with partial lipodystrophy, who started with a lower leptin level had a better response, but some patients with higher leptin levels still responded to leptin therapy with an improvement in hemoglobin A1C. So this suggests that plasma leptin levels might in time be a useful diagnostic tool for determining whether or not a patient with lipodystrophy is likely to respond. On slide 35, you'll note that 3 patients with partial lipodystrophy with very different appearances. The 2 women on the woman on the left and the center actually would be not obvious as having lipodystrophy, but they do. And so plasma leptin levels might be useful for helping to predict a biological response. Now, there are other forms of lipodystrophy. On slide 36 is shown a patient who has an acquired form of lipodystrophy associated with HIV, and HIV lipodystrophic patients have a partial form, and as shown on slide 37 is a research report showing that leptin treatment also can be effective for this condition. Now, I want to now move to slide 38 to point out something about lipodystrophy that might suggest that there are polygenic forms as well. For those of you who practice clinical medicine, the patient on slide 38 looks similar to many other diabetic patients you might see in your clinic. Central obesity and skinny arms and legs. On slide 39, I refer you to a research report describing what's referred to as the metabolically obese normal weight individual. And it's pointing out that many diabetic patients actually have what looks like an appearance consistent with lipodystrophy. This has raised the possibility, as shown on slide 40, that lipodystrophy might be a spectrum disorder, extending on one end from as monogenic and recessive acquired complete forms, but that there might be polygenic forms in the general population at the other end, which might be these so-called metabolically obese weight normal patients. And indeed, on slide 41 are 2 recent reports of genetic studies in humans that suggest exactly this point. In both these studies, which led to the same conclusion, they clustered diabetic patients into different groups based on a set of polymorphisms. On slide 42 is shown one of the clusters. It's the cluster on the left. Many of the diabetic patients, about 7% of the diabetic patients in this cohort, had this cluster of abnormalities, and the investigators found that that group of individuals tended to be thin. Moreover, if they look, as you can see on slide 43, at the clinical parameters of these patients, they all showed signs and symptoms characteristic of lipodystrophy. Now, I went through this very quickly, but the point of it is that, as shown again on slide 44, the data suggest that there are likely to be polygenic forms of lipodystrophy, and that indeed lipodystrophy is a spectrum disorder. And one might wonder whether the patients with this polygenic form who were severely enough affected and had low leptin levels might also respond to leptin therapy. Now, this so-called polygenic form of lipodystrophy is not rare in the Western world, but it's actually somewhat common in Asia, as shown on slide 45, where the metabolic syndrome is often associated with relatively low BMIs in especially South Asians. So on slide 46 is a brief set of points I'd like to make now. This will tee things up for Dr. Unger in a few minutes, but it points out that leptin can treat diabetes in lipodystrophy, and in animal studies it can even do so in the absence of insulin signaling. And so in some cases, as I'll show you in a moment, mutations of the insulin receptor result in insulin resistance that's treated with diabetes. Now, the mechanism appears to be a result in large part of a suppression of hepatic glucose output, that, though Dr. Unger will talk more about this, and leptin can also increase glucose turnover and metabolism even in normal animals that are studied. On slide 47, as promised, is a reminder for me to tell you that leptin markedly reduced insulin levels in the most insulin-resistant syndrome there is, Rabson Mendenhall Syndrome. These patients have insulin receptor mutations and show a marked improvement in glucose and insulin on leptin therapy. This suggests that leptin might also be a potential use in the context of other alterations of insulin signaling. And I'll point out to you that PI3 kinase and AKT inhibitors are now being developed for cancer. So on the next slide is a very brief summary of what I tried to communicate thus far. There are leptin deficiency syndromes that can be treated with leptin. This includes patients with mutations in the leptin gene as well as lipodystrophy. I didn't show you the data, but there are also reason to believe that leptin replacement can be also useful for hypothalamic amenorrhea and other conditions. In the remaining 3 or 4 moments, I just want to now segue and tell you about leptin and obesity. On slide 49 are data showing you that obesity in the general population is not a leptin deficiency syndrome as compared to lipodystrophy. In that slide, what you can see is that as body fat increases, leptin concentration in blood increases. And this is suggested, as shown on slide 50, that obesity is a leptin-resistant state. What this means is, analogous to type 2 diabetes and insulin resistance, is that in obesity, the leptin the body makes doesn't work well enough. There's an apparent block to leptin action. As a consequence, weight increases and leptin level increases, resetting weight at a new higher leptin level. On slide 51 are some early data suggesting that leptin might be useful for the treatment of obesity, and this data showed on slide 51 that the highest doses of leptin could reduce weight in some patients, but not all. But this was too high a dose to be delivered commercially, and so leptin therapy at lower doses was not effective, and its use for this condition was not pursued further. However, more recently, it's been suggested on slide 52 that there might be a responder subset of obese patients. Certainly a subset of obese patients do respond to leptin therapy, and there's been reason to believe that there might be obese patients with relatively low leptin levels and that they would respond better. And in fact, there has now been an early proof of principle study by Elif Oral at Michigan showing, in fact, a clinical benefit among obese diabetic patients with low leptin levels. I'll close by telling you on slide 53 some of more recent research from our laboratory and others that seeks to identify the molecular basis of leptin resistance. On slide 54, I just point out to you that understanding the basis of leptin resistance will require that we understand more about the neural mechanisms by which leptin acts and what's wrong with those actions in the leptin-resistant state. In slide 55 is a diagram showing that we now know that leptin acts by modulating the activity of nerve cells that regulate appetite. Leptin inhibits nerve cells that stimulate feeding. An example of this are neurons that express leptin receptor NPY, shown in green. NPY is a neuropeptide. At the same time, leptin inhibits neurons that activates neurons that inhibit feeding. An example of this are neurons that express leptin receptor and MSH, shown in the red. And on slide 46 is a slide showing you that mutations in this neural pathway cause human obesity. We now know that 10% of morbid obesity is a result of mutations either in leptin, the leptin receptor, MSH, or its receptor, the MC4 receptor. Well, in the interest of time, I'm going to close and ask you to skip slide 57 and move on to slide 58, which simply explains that we now know that body weight is regulated by a classic feedback loop comprised of leptin as an afferent signal, brain centers that control appetite and metabolism that act by regulating appetite, as well as a series of end organs, and that this has implications for diabetes. Certainly, it's an effective therapy for lipodystrophy, and pending further studies of its efficacy and safety might be useful for other forms of diabetes as well, as you'll hear in a moment. So if you turn now to slide 59, I'll just summarize what I hope to have communicated. One, leptin is an approved therapy for generalized lipodystrophy. It has beneficial effects in some partial lipodystrophy patients, but is not yet approved for this indication. Leptin does not necessarily require the action of insulin, and leptin has limited efficacy for most type 2 diabetic patients who are obese, but it might have benefit in a subset who start with low leptin levels. So with that, I'll stop and hand the podium over to Dr. Unger, and I'd be happy to take any questions you might have at the end of his presentation. Thank you for listening.
Great. Thank you very much, Dr. Friedman. That was a very interesting presentation. Now, I'm also honored to introduce our next distinguished speaker, Dr. Roger Unger, also a physician scientist. Dr. Unger is a professor of internal medicine at the Dallas VA Medical Center and is the Touchstone West Distinguished Chair in Diabetes Research at UT Southwestern Medical Center. In 1959, at the Dallas VA Hospital, Dr. Unger introduced the Glucagon radioimmunoassay, which was the first radioimmunoassay to be reported. After the insulin radioimmunoassay was published the following year, the Unger lab became the first to examine the interacting role of both hormones in physiology and pathophysiology. By 1975, they had reported that diabetes was characterized by hyperglucagonemia, and that when glucagon was suppressed by somatostatin, the hyperglycemia disappeared. For 30 years thereafter, Dr. Unger searched for a glucagon-suppressing agent that could be safely used to treat type 1 diabetes, which led to his work on leptin. Dr. Roger Unger has received countless honors and awards, including the Banting Medal from the ADA, the Claude Bernard Award of the European Society for the Study of Diabetes, the J. Dennis McGarry Award for the Scientific Excellence in Diabetes, and was elected to the National Academy of Sciences in 1986 and the American Academy of Arts and Sciences in 1993. We are very excited to have him with us today as well. So with that, I turn it over to Dr. Roger Unger.
Thank you very much. If I were to be asked what the two most important diabetes-related publications of the 20th century are, I would pick the one on top, which is a no-brainer. Everyone in the world would pick that one. Second one might raise some eyebrows. I'm not sure if the second paper even mentions the word diabetes, but probably does. In any case, it has not captured a huge therapeutic market in diabetes, and except for the specific effects in leptin deficiency states, there's no real evidence that it has a place in treating common forms of diabetes. So I'm tempted with the South Asian syndrome of low fat, probably low leptin levels. It would seem to me that some of those patients might be responsive. In any case, the euphoria that followed the discovery of insulin and its use to treat type 1 diabetics in ketoacidosis captured the imagination of the whole world. And The idea was that if you took insulin, since insulin saved lives, the life that it saved would be a pretty normal one. This turned out not to be the case. Normal people, as shown in the left-hand panel, in an article by Sawdak at Johns Hopkins, shows the remarkable glucoregulation of non-diabetic individuals in whom any glucose load or any glucose need will not deflect glucose levels above or below the narrow range shown in the slide. In contrast, even optimally controlled type 1 diabetics, and these are the model patients from Sawdak's clinic with a mean fasting glucose of 121. Even such well-controlled patients show wild fluctuations in glucose, sometimes reaching towards 400, interspersed with plunges down as low as 25 and below. This is unique in clinical medicine. It deserves to carry the name of a syndrome, such as glycemic volatility. But to the discredit of all of us in the field, clinicians, scientists alike, this frightening, hectic behavior of an important homeostatic entity has been pretty much shifted to the patient. Instead of trying to find out why this occurs and how it can be prevented, we tell our patients, if you want to be well controlled, check your glucose levels 7-8 times a day and adjust your insulin and food accordingly. And yet, years of evidence by many different labs have led to accumulation of information that really would explain the mechanism of this syndrome. Let's start with the microorgans that regulate glucose and nutrient metabolism, the pancreatic islets. In this cartoon taken from Bosco, it demonstrates what Lelio Orci showed in the late 50s, that the islets have very closely related insulin and glucagon components. Not only does 90% of the alpha cells have a contiguous beta cell neighbor with which it shares an interstitium, but the blood vessels carry insulin from the beta cell to the alpha cell. And finally, the two cell types have gap junctions, which make them into a syncytium, small molecules passing from the cytoplasm of one cell to that of another. In order to find out what this geographical, these are experiments designed to understand the functional implications of the juxtaposition of alpha and beta cells. And in these studies in normal rats, glucose was perfused into the pancreatic artery at high concentration, stimulating the insulin, and has found this lowered the glucagon levels below 1,000 picograms per ml in the venous effluent. At 10 minutes, a potent anti-insulin neutralizing serum was perfused, and within seconds, a rise in 150% rise in glucagon began. This told us that the contiguity of the two cell types was designed to provide the alpha cells with a powerful glucose sensor, the beta cell, which in turn can control the secretion of glucagon and produce a glucagon insulin ratio appropriate for the moment. No, that's not on slide. Slides are all mixed up here. Now, in the non-diabetic, then, the beta cells secrete insulin, which provides the alpha cell with the highest insulin concentration anywhere in the body, which we estimate at between 2,000 and 4,000 microunits per ml. This creates an insulin-glucagon ratio that's appropriate to the condition of the moment. The two hormones leave the pancreas and form a mixture, a bihormonal mixture that reaches the liver. 90% of the insulin receptors are in the splanchnic compartment and virtually all the glucagon receptors, so that the post-hepatic levels of the hormones are rather unimportant. The low levels of insulin, however, are sufficient to inhibit lipolysis in adipocytes. Now, when we try to mimic this in a type 1 diabetic, obviously we're putting the insulin in the wrong compartment. If it's injected subcutaneously, it will take probably at least an hour before most of it's absorbed. And when it's absorbed, it'll go around the systemic circulation. There's no preferential accumulation of insulin in the splanchnic compartment where most of the receptors are located. On the other hand, glucagon has a straight shot at the liver with no dilution at all. This makes it very hard for the injected insulin to create a high enough insulin-glucagon ratio to do what we intended it to do. Because it was so difficult to raise the insulin-glucagon ratio adequately by peripheral injections of insulin, we thought another way of raising the ratio would be to lower the denominator. And fortunately, Roger Guillemot at the Salk Institute had discovered the hormone somatostatin, and it was shown in Seattle to be a relatively nonspecific inhibitor of exocytosis by endocrine and exocrine secreting cells. So we wondered what would happen if we could suppress glucagon. And in those days, there was no IRG. So instead of using rodents, we used type 1 diabetic patients. And this study was done primarily by Dr. Philip Raskin. And what he did was reduce the insulin level that these patients were taking by 90%. And when he did that, glucagon rose, as did glucose, hovering in the 300 range, and glycosuria of 50 grams per day appeared. When somatostatin was started, glucagon was suppressed, and glucose levels followed, coming down to normal or near normal levels, and the glycosuria was eliminated. Now, this was a very gratifying result, but we had to quit because there were some undesirable side effects that precluded continued use of this agent. And there being no other known glucagon suppressor available, we left the field completely and turned our attention to other matters, in particular the matter of obesity and ectopic metabolic syndrome due to ectopic deposition of lipids. And we observed that accumulation of fatty acids could produce the manifestations of type 2 diabetes. And within 1 month of publication of our paper on lipotoxicity, the Jeff Friedman paper announcing the discovery of the OB gene appeared, and we wondered if this might be a means of reversing the lipid accumulation in the islets. We therefore did this experiment. We created a type 1 diabetic rodent, streptozotocin, and after the diabetes appeared, we injected islets from normal, closely related donors and injected those islets into the liver. The reason we did this is we thought that the high insulin coming out of the islets would turn on lipogenesis in the surrounding hepatocytes, causing a triglyceride halo. The high levels of lipoprotein lipase in the beta cells would hydrolyze the triglycerides and flood the beta cells with excess fatty acids, which we thought would do damage. And this is exactly what happened. The beta cells were killed, triglycerides accumulated in the cells, and we called Dr. Friedman to see if he could spare some recombinant. It still hadn't been named leptin in those days, but that's what we wanted. He explained that it was not possible at the moment to supply the protein, so we decided to express leptin using an adenovirus. So we injected the diabetic animals before transplantation, either with an adenovirus containing the leptin cDNA or the adenovirus alone. And we gave a suboptimal dose of islets, which caused some improvement in the unleptinized animals. But in the leptinized animals, there was normal glycemia. And for a brief moment, we thought we had cured type 1 diabetes because doing this, giving leptin together with islet transplants, should give us a normal glycemia, at least for a few weeks. However, our joy was short-lived because we did another control experiment. We gave the virus with the leptin, but we did not give islets. And we got just as good normal glycemia without any islets as with islets. And that told us immediately we must have suppressed glucagon. So we did experiments in which hyperleptinemia was produced adenovirally, and type 1 diabetics were followed for several weeks. And as you can see, they were all normal glycemic. We actually carried some up to almost a year. They remained in good condition with no insulin or C-peptide in their body, but as the 1-year anniversary approached, the glucose levels creeped up into the low 200 range. But they never got type 1 diabetes again. They never got insulin-deficient diabetes. So now we thought we had more insight into the cause of the hypervolatility and how to deal with it. We created a murine model of hypervolatility, which is even more volatile than human type 1 diabetics. And we examined the hormone levels in the minimum when the glucose levels were low and when the glucose levels were high. When the glucose levels were low, insulin measured 20 times the levels of non-diabetics at the same time of day. So that was a no-brainer So we reduced the insulin, and when we did that, the insulin levels became normal, as did and the hypoglycemia disappeared. But when we reduced the insulin, the glucagon shoots up. This went to 5 times normal, and with it, hyperglycemia gets worse. So now we wanted to use a glucagon suppressor. At this time, recombinant leptin was available. We gave it to the animals, even though we had no insulin. Glucose levels in these type 1 diabetic mice were perfectly normal. So we used, what we did then was we treated a large group of NOD mice with Islet pumps containing either leptin or just buffer. The blue squares show you the insulin-treated animals with the volatility of their glycemia, and the yellow squares show the leptin-treated animals with no insulin whatsoever. So it turned out that, what you should be seeing is a suppressed glucagon level in the mouse on the left. Mouse on the right has a very, very high glucagon level, and I think you can notice that there's also a highly significant p-value in its sawdust. Based on the remarkable effects of leptin treatment in the place of insulin, we decided to try to learn more about this relationship between the two hormones and what they really do in metabolic regulation. We were beginning to question the validity of the insulinocentric ideas that we were all taught early in our careers. And fortunately, we were able to obtain a colony of glucagon receptor knockout mice from Marine Sharon at Albert Einstein. And these mice arrived in Dallas. We looked at receptor of the glucagon, expression of the glucagon receptor, messenger RNA in their livers, and they all were undetectable. All of the animals had normal glycemia. So at 7 or 8 days, at 2 days, we gave them streptozotocin in very high doses, intending to destroy all their beta cells. Their insulin and C-peptide levels were undetectable, and those that were looked at autopsy, we found less than a fraction of 1% of insulin-positive cells remaining. Treatment of wild-type animals with that dose of streptozotocin invariably led to severe type 1 diabetes with ketonuria and death within 2 weeks unless they're treated with insulin. So these animals were perfectly normal. Not even the glucose tolerance was abnormal in these streptozotocinized animals with no glucagon receptor. So we decided to give them a glucagon receptor. And at 7 days after the streptozotocin, we injected them with adenovirus containing the cDNA of the glucagon receptor. And the messenger RNA of the receptor shot up transiently and then disappeared at 15 days. Within 2 days of the appearance of the glucagon receptor, hyperglycemia appeared and went up almost to 500 before it subsided with disappearance of the receptor. So this made us wonder who was in charge in these islets. We certainly know that the beta cell is the glucose sensor, but it looked to us as if without glucagon to control, the insulin had very little to do. Next slide. So we looked at another way of getting rid of the glucagon action without knocking out the gene. We obtained from a company called REMD Biotherapeutics a glucagon receptor antibody, and we treated type 1 diabetic rats and mice with the antisense. Next slide. The glucose levels were reduced to below 100 milligrams per deciliter in every animal and remained normal for up to 10 days. After 10 days, they drifted back up, but another shot of antibody, 5 milligrams every 10 days, kept the glucose normal. Next slide. Now I want to change from type 1 diabetes to type 2 diabetes. Totally different disease. The only thing they have in common is their name and their hyperglycemia. If you look at the islets, normal islets, type 1 diabetic islets, you see that type 2 is quite different. Instead of the near total deficiency of beta cells, the beta cells are abundant. The red staining cells are full of insulin, and they're probably, at least in the early stage of the disease, more than in the normal state. Yet hyperglycemia is present, obviously, and glucagon levels are very high. Why would the glucagon levels be high when there's so much insulin? Well, it turns out that the alpha cells, like so many other cells in type 2 diabetes, are insulin resistant. But irrespective of the cause of the hyperglycemia, it appears that glucagon action is necessary to maintain it. So here again, in DB/DB mice with severe type 2 diabetes, administration of the antibody completely normalizes their glucose levels. So we, as much as this offends us, our conventional thinking, what we were taught about diabetes, we have to face the facts as they emerged from these studies, namely that the insulin-glucagon ratio sets the appropriate metabolic mode for varying caloric environments. When the beta cells are absent, or when the alpha cells are insulin resistant, hyperglucagonemia lowers the insulin-glucagon ratio and diabetes appears. We have never identified any form of diabetes in which glucagon was not high relative to insulin. And this includes total pancreatectomy, where gastric fundus alpha cells are hypersecreting glucagon. Thirdly, diabetes can be repressed by blocking or suppressing the denominator of the insulin-glucagon ratio as easily and possibly more, even more easy than by increasing the numerator. And finally, here I hesitate with this fourth conclusion. We want to test this far more extensively. Insulin's actions seem to be mediated by glucagon. And without glucagon, we have trouble identifying any insulin action whatsoever. But we're still looking. with that, I'll stop and answer questions. Sorry the slides were so mixed up.
No problem. Thank you, Dr. Unger. We really appreciate your presentation as well. In the essence of time, I think we'll just do two quick questions. The first is, can you comment, either one of you, on the premise of including adiponectin testing in conjunction with leptin when assessing individuals for metabolic syndrome or other indications of insulin resistance?
Yeah, well, you know, in addition to the actions of leptin that were described by Dr. Friedman, we have suspected there might be some other very important functions of leptin in obesity. The fact that in diet-induced obesity causes a rise in leptin raises the possibility that that is doing something beneficial to the overnourished, overweight individual. And what could that beneficial thing be? Well, we know that leptin is a powerful activator of AMP kinase, so it might be upregulating the oxidative machinery of cells that do respond to it. As Dr. Friedman pointed out, the adipocytes don't respond to it, to their own leptin. There's no autocrine action during the obesity, but the non-adipocytes do. And in experiments that I don't have time to show you, we are able to lower ectopic lipids with leptin, so that we think that there's a place for leptin treatment in certain forms of overnutrition when there is evidence of accumulating ectopic lipids.
Thank you. Dr. Friedman, any comments?
Well, I would just say that I'm not sure, for the specific question about measuring adiponectin, I'm not sure what actionable information would be derived from that. There was some speculation that in lipodystrophy, a lack of adiponectin might contribute to the phenotype, but based on some experiments I could explain if there's further interest, I don't think there's very much evidence that adiponectin, well, low adiponectin has anywhere near as much to do with a lipodystrophic phenotype than does a lack of leptin.
Okay, thank you. We will take one more question, and it is, do you think leptin therapy could have effects in gene expression? If yes, which genes do you think are involved?
So I'll take that. So there's absolutely effects of leptin on gene expression. Some of the effects on gene expression are in the central nervous system. So leptin can change the activity and the gene expression profile of specific nerve cells in the brain. These in turn have effects on most other organ systems, and there are so very dramatic changes in gene expression in fat, in liver, and elsewhere, and this is part of the way that leptin acts.
Okay, so in the essence of time, I think we need to stop here. I'd like to thank our two speakers for excellent presentations and thank everyone for joining us. Hope you enjoy the rest of your day.